Well, we brought our precious Muck samples back to the Genspace Community Lab in Brooklyn. The first step is to take a very small sample out of the tubes and put it into a little tiny test tube that has little beads in it, and we put it on this mixer that vibrates, and the beads rattle around in the tube and break apart any bacteria, any spores, any hard-sided little creatures that might be in there, so we can extract the DNA.
So it's from this sample of muck dredged up from the canal. So after you centrifuge it, all the little bits of debris go to the bottom. And then we just take the part off the top that's the purest. Hopefully that's going to contain our microbial DNA.
So we got a little surprise when we opened up the jars. We found a whole bunch of these little tiny red worms that were trying to escape out of the top. So they had crawled up out of the muck, so we pulled a few out. But I don't know what species they are, and they're really kind of creepy. When we first looked at them, they were pulsing. They were actually pulsing and moving. It's kind of like the beginning of a horror movie, you know? You take the sample out and something crawls out of it, and that's kind of the beginning of the end. At any rate, we're going to do some DNA barcoding on these and figure out what species they are. There's really a potential for finding something that's unique, because these are extremophiles.
Right.
They're living in an incredibly polluted environment, so they may be metabolizing things. Maybe there's an antibiotic to be found. Maybe, who knows what sort of metabolic pathways these things have, or what they've mutated into. And who knows, we may discover a new life form and get to name it.
That would be awesome.
So gel electrophoresis is kind of like making DNA run an obstacle course. You have a matrix that the larger DNA molecules have a harder time squirming through than the smaller ones. DNA is a long polymer, so it's like a long thread. And so the bigger pieces take longer to squirm through than the smaller pieces. And the reason they're moving is because you put an electrical current across it, and DNA has a net negative charge. So those bright bands that you see are actually the DNA from the samples running through the gel matrix. That's how we know we have DNA. We have DNA! In every sample we have DNA. It's great.